Assalamu alaikum. Dear students, this is me, Atiq Ahmad. Today we are going to study the topic karyokinesis inside mitosis. We will have an overview of the previous lecture to connect the previous knowledge with the current one. During the previous lecture, we have studied the division phase. And before that, we have studied the interphase. Now, broadly speaking, the cell cycle consists of two major stages. The interphase, which is the preparatory phase, which we had already studied. Then in the previous lecture, we have studied the second major stage, that is the division phase. The division phase again consists of two types of divisions: mitotic division, mitosis, and meiosis. So in the previous lecture, we have started the mitosis. Now the mitosis consists of two major stages: the karyokinesis and the cytokinesis. The karyokinesis is the division of the nucleus, and the cytokinesis is the division of the cytoplasm. So inside mitosis, today we will study the karyokinesis, and then in the next lecture we will study the cytokinesis. So we will start from the karyokinesis of mitosis. So karyokinesis is the division of the nucleus, as we have already studied in the previous lecture too. Karyokinesis is the division of the nucleus. Today we will study the four phases of the karyokinesis. Karyokinesis is further divided into four phases: prophase, metaphase, anaphase, and telophase. We will study all these four phases one by one. We will start from the first phase, that is the prophase. Now, before proceeding to this topic, I will have a summary of the prophase. The prophase basically consists of four major events. What are those events? In the prophase, number one, the chromosome will condense, and proper chromosomes which are visible under microscope, that will be formed. This is the first event. Secondly, the nucleolus will disappear. Third, the spindle fibers will be formed. And fourth, the nuclear membrane will break down. These are the four changes which will occur in the prophase. Now we will move towards the detail. Chromatin threads condense, coil, and shorten to become chromosomes. Now recall, in the interphase, the chromosomes are in the form of thread-like structure. Those threads are known as chromatin. That network of threads are known as chromatin. So the chromatin will condense, coil around the histone proteins, and will shorten to become chromosomes. So the threads will coil around the histone proteins and proper the Y shape, this shape, chromosome will be formed. Each chromosome consists of two sister chromatids attached at the centromere. Now this is a chromosome, this is one chromatid, this is another chromatid. They are attached here at the center, this central region, this central region, this one, this central region, this is known as centromere. The chromosomes are now visible under microscope because now the chromosomes are in condensed form, so they are visible under the microscope. While on the other hand, in the interphase, which we have already studied, in that phase the chromosomes were in the form of chromatin network, and that was not visible under the microscope. But now it is visible under the microscope. Now we have studied the early prophase. Now what will happen in the late prophase? In an animal cell, the two pair of centrioles reach to the opposite pole of the cell. They make a network of spindle fibers between the two poles. Now just recall, at the end of interphase, in the previous lecture, in the at the end of interphase in the G2 phase, the centrioles are duplicated and they start moving toward the opposite poles. Now in prophase they have reached to the opposite poles, and now they will form spindle fibers. So in an animal cell, the two pairs of centrioles reach to the opposite pole of the cell. Now they have reached to the opposite pole of the cell. They make a network of spindle fibers between the two poles. They will form spindle fibers from the two poles. The complete set of spindle fibers is called as mitotic apparatus. That complete apparatus is known as mitotic apparatus. Now these are the chromosomes which are formed. Now this is one chromatid, this is another chromatid, and the central region, this is the centromere. The central region is the centromere. Now in another diagram it is very clearly shown here. In interphase the chromosomes are in this chromatin form. Now in prophase the chromatin is coiled and condensed and shortened, and these chromosomes are formed. Now look at these chromosomes. This is one chromatid, this is one chromatid, this one, and this is another chromatid. This is one chromatid, this is another chromatid. They are attached at the central region. This central region, this is the centromere. So this is one chromatid, this is another chromatid, and the central region, this white one, this is the centromere. This is the centromere. And from these centrioles spindle fibers will start. So these spindle fibers, these are shown here, these spindle fibers are formed. So spindle fibers will be formed. The complete set of spindle fibers is called as mitotic apparatus. In plant cell, there are no centrioles. We have already studied in chapter number 4 that the single organelle which is not present in the plant cells, that is the centriole. So in plants there are no centrioles. So what will happen? How spindle fibers will be formed? Because the centrioles are responsible for the formation of spindle fibers. So in case of plant cells, the cytoplasm basically have the spindle fibers, and those spindle fibers will aggregate, they will combine together to form a network which is mitotic apparatus. So in plant cell there are no centrioles. Their mitotic spindles is formed by the aggregation of spindle fibers present in the cytoplasm. In the cytoplasm, spindle fibers are present. They will combine together to form spindle fiber for the mitosis. Now as a part of the changes of prophase, the nucleolus will disappear. I have already discussed, and the nuclear envelope will break down. I have already discussed. So to sum up, the four changes, or in other words the four events which occurs in the prophase of the mitosis, those are formation of chromosome, condensation of chromosomes, proper chromosomes will be formed. Secondly, the spindle fibers will be formed. Thirdly, the nucleolus will disappear. And fourthly, the nuclear membrane will break down. These are the four changes which occur in the prophase of karyokinesis inside the cell. Now we will be towards the metaphase. What happens in the metaphase? The chromosomes which are already formed, they will they will arrange themselves at the center of the cell, and the spindle fibers, two spindle fibers from this side and two from this side, they will attach with the chromosome at the center, at the centromere position. Now this is described here. In metaphase, some spindle fibers attach with chromosomes. These are the spindle fibers, these from this side as well as from this side. So in metaphase, some spindle fibers attach with chromosomes. Two spindle fibers from both sides attach with one chromosome. This is one chromosome, this is another chromosome. These two spindle fibers from one side, two from another side, they will attach with this chromosome at the centromere position. The chromosome attached with the spindle fiber arrange themselves at the equator of the cell. They will arrange themselves at the equator, or in other words, in the central region of the cell. This is the central region of the cell, equator of the cell. In this way, the plate which is formed, that is called metaphase plate. So here, this region where the chromosomes are arranged, here a metaphase plate will be formed. So this plate is known as metaphase plate. The chromosomes are arranged at the central region, at the equator of the cell, of the nucleus. Now anaphase. During the anaphase, what will happen? The spindle fibers which are already attached with the chromosomes, they will pull the chromosomes toward the opposite end. But before that, the centromere will split, will break down. As a result, the two chromatids will be separated. Now we will consider those chromatids as daughter chromosomes, and they will be pulled toward the opposite poles by the centrioles. So the details are here. Early anaphase, and then we will say the late anaphase. The centromere of each chromosome splits. Splits means break down. The spindle fibers pull the chromatids apart to opposite poles. Once the chromatids are separated, they are called daughter chromosomes. Now here it is shown. The chromosome, the centromere will split. The two chromatids will be separated. Now these are called daughter chromosomes, and they are pulled by these spindle fibers toward the opposite poles. Now they start moving towards the opposite poles because the spindle fibers pull them towards the opposite poles. In early anaphase, the centromere split to the other pole. Half of the chromosome will move toward the one pole and half to the other. In the late prophase, the chromosome have almost reached to their respective poles. Now this is the late prophase. The chromosome have almost reached to these poles. This is one pole, this is another pole. These are the chromosomes. They have reached to the opposite poles. The cell membrane begins to move inwards at the center. Now at this stage, this is the cell membrane. It will move onward. This is inward, and this is also moving inward. Now telophase. This is the fourth stage, the last stage, telophase. In telophase, spindle fibers break down. A nuclear envelope forms around the chromosome at each pole of the end. Now to sum up these all things, just remember a simple key. The four changes which were already taken place there in the prophase, that will be reversed. In the prophase, what happened there? In the prophase, chromosomes were condensed. Now the reverse will occur here. They will again be converted to the chromatin network. They will uncoil and they will form chromatin network. Now in prophase, we have already studied that the nucleolus will disappear. Now in telophase, the reverse will occur. The nucleolus will reappear. Thirdly, the nuclear membrane was broken down there in the prophase. Now the reverse will occur here in the telophase. The nuclear membrane will be reformed. And number 4, the spindle fibers were formed there in the prophase. Now those spindle fibers will be broken down. So these 4 changes which already occurred in the prophase, those 4 changes will be reversed here in the telophase. Now let's move into the details. In telophase, spindle fibers break down. In prophase, it was made and now they will be broken down. A nuclear envelope forms around the chromosome at each pole of the cell. Thus the 2 daughter nuclei are formed, each with the same number of chromosomes as were present in the parent nucleus. Because this is mitosis, so the daughter nuclei will have the same number of chromosomes as that was present in the parent cell. A nucleus forms and the chromosome uncoiled to become thin chromatin network. Now just focus on it. The 4 changes which has already took place in the first step in the prophase, those all are reversed here. Spindle fibers are broken down, nuclear membrane will be reformed, nucleolus will reappear, and the nucleolus will reappear. So the 4 changes, and the spindle fibers will be broken down. So these are the 4 changes which occur here in the telophase. As a result, 2 nuclei are formed. Now diagrammatically, in another diagram it is shown here. This is interphase chromatin network. Here proper chromosomes are formed, condensed form, spindle fibers are, that is shown here, but it should be shown here. Spindle fibers are formed. Now here this is the equatorial plate. So in the metaphase, this metaphase plate is formed. The chromosomes are arranged at the center of the cells, and the spindle fibers attach with the chromosome through the centromere. This is the anaphase. The centromere split and the chromosome moves toward the opposite ends, and this is the telophase, where the nuclear membrane will reappear, the nucleolus will reappear, the spindle fibers will be broken down, the nuclei will be divided into 2 daughter nuclei. This is all about today's lecture. Inshallah, see you next time with the new lecture. Take care. Allah Hafiz.